Alright, hi guys, this is a video review of lectures 10 through 12 of Physiology. Let's get started. So here I wrote out all the different equations for these lectures. Not going to go over them right now, so I'll go over them in just a minute, but here they are, so we can come back to them later. Alright, so primarily we're focused on fluid flow, meaning how the blood is going through the different vessels throughout the body, and then the equations that describe them, as well as really regulation of fluid flow. Making sure that we're getting blood where it needs to go so that the body can get the nutrients it needs. So really starting with discussing control of fluids, we have acknowledgement that the heart is acting as a pump and providing pressure, and then the vessels throughout the body are providing resistance. And so really, the first place to start here is talking about the flow of fluid. And so the heart is providing pressure, the vessels are providing resistance, and the flow, Q, is caused by the pressure and limited by the resistance. And so that gives us our first equation, which is Q is equal to delta P divided by resistance. So flow is caused by pressure and limited by resistance. And then just like conceptually thinking about what's going on with resistance, and specifically in the lectures that bring up the point of parallel versus series resistance, like just imagine, you're to think about resistance as a property, you're trying to breathe through a very tiny straw, just one. It's really hard. You can't breathe through your nose. And then somebody hands you a second straw, and now you have two tiny straws to breathe through. Isn't it a little bit easier to breathe through, right? So basically, with parallel resistance, the more pathways you have for blood to flow, more capillaries, for instance, you have less total resistance. That's like breathing through two tiny straws, easier to do than breathing through one. So fluid flow would be dependent on parallel versus series, and then also the different parameters of the pipe that the fluid is going through, right? And so the other example to think about is, I know when I was a kid, we used to, at camp, play this game where we'd suck Jello through a straw and see who could do it the fastest. They've gone to some strange camps, but anyways, imagine you're trying to drink Jello through a straw, and then ask yourself, like, what's easier, a shorter straw or a longer straw, right? If it's a really short straw, it's pretty easy. If it's longer, it's harder, right? If you've ever tried to drink a Frosty through a straw, it's like really hard to do, but the straw's really short, it's not too bad. All right, a wide straw versus a thin straw. Well, a wide straw is going to be easier. And then, would drinking water through that straw be easier than drinking Jello? This is, right, viscosity. Something with low viscosity like water versus something, with high viscosity, difficulty of flowing, like Jello or honey, is going to be harder. So that brings us to all the different components of, and mispronounces this law's name, but Poiseuille's law, which is the resistance through a single pipe is equal to 8 times the viscosity times the length, right? Increase in length means more resistance drinking through the longer straw. Increase in viscosity means, just like trying to drink Jello through a straw versus drinking water. That's more viscosity means more resistance. And then, the thicker the straw, wider diameter, bigger cross-sectional area is on the bottom, so pi r to the fourth. All right, so that brings us to, that's our second equation we need to know. And then just going back to this equation up here, the total flow through system is equal to the total change in pressure divided by the total resistance. And so an individual pipe's resistance is based on Poiseuille's law, or individual vessel. But as we said, you could have multiple straws in parallel, just like we have with the actual human body, where we have, multiple arterioles or multiple capillaries in a bed. More than one pathway will yield a lower resistance than just one pathway. And so the equation for vessels in parallel is R total, or 1 over R total, is equal to 1 over R1 plus 1 over R2 plus 1 over R3. And then if you do some math manipulation, algebraic manipulation, you get this equation. R total is 1 over R1 plus 1 over R2 plus 1 over R3 plus however many resistors or vessels you're concerned with. All right, that brings us to, putting things in series. So imagine you have, one tiny straw that you're trying to breathe through. You're like, this is pretty hard. And then somebody gives you, a second straw, and you're like, okay, you're like, oh, but it's bigger. Shouldn't this be easier? Well, no, because there's still, the net straw is longer, right, than it was before. You still have all the resistance of this straw, but now you have some surface area on the second straw that's been added to it for, the air molecules to bump into. And so adding more and more resistors, and then someone adds a third straw to the end of the second straw, if you add our vessels in series to each other, the total resistance is just going to add up one after another. So that's our fourth equation. R total is equal to R1 plus R2 plus R3, and this would be like our artery to our arteriole, in line with our capillary, in line with our venule, in line with our vein. All right, so as we get further and further, the total resistance is increasing. All right, so then looking at individual vessels, right, we can see that the aorta has an enormous diameter, and then specifically the capillaries have the tiniest diameter, 3 micrometers, which is minuscule. And so it's interesting to know, and we're gonna make this point at least twice, the capillaries have a small individual cross-sectional area, and so you're like, that's the tiniest straw to drink through, right? So capillaries have the smallest individual cross-section, but they have the greatest total cross-section when you look at all the capillaries together, right? So this is important to keep in mind as we talk about, the net system of the human body, which is, right, if you have two pipes, two arterioles, and they're in parallel with each other, is this going to be easier to have fluid go through, or would 10,000 individual capillaries, right, the sum of 10,000 individual capillaries diameters together is more cross-sectional area than, right, like 2 arterioles. So the total cross-sectional area of the two capillaries is the greatest, and that has some effects when we look at our system of the human body as a whole overall. So we'll get to that in just a minute. We were given this diagram right here in lecture, and there's a couple of things to point out. So we have the aorta on the side, and then just noticing that the aorta has the highest pressure, right? That kind of makes sense because the left ventricle is pumping directly into the aorta, so it should have the highest pressure. And then the velocity is also greatest there and drops off. And so velocity for fluid flow, you have an intuitive sense of this. It's greatest in the aorta right after it's been squeezed out of the ventricle, the left ventricle pump, but it increases with less cross-sectional area, right? And so less cross-sectional area, think about it as, when you're a kid, presumably you play with a hose, right? And our hose is getting the fluid to flow out, say, to 5 meters per second, and then you go ahead and you stick your thumb on top over the edge of the hose, and you're decreasing the cross-sectional area. And so as a result, right, the fluid just sprayed out of that hose really quickly, and so the net velocity increased, right? And so if you have less cross-sectional area, the velocity is going to increase overall for the same fluid flow, right? And so the idea is that the velocity will increase with less cross-sectional area, and we said that the capillaries, despite individually having the least cross-sectional area, because they're the smallest, they are going to, in total, have the greatest cross-sectional area, right? And so you can see the cross-sectional area here spikes where you have the capillaries, and that coincides approximately with the lowest velocity. And then as you get to the less numerous vessels on either side, you can see that the velocity increases as you have a net less cross-sectional area, right? But think about it more like this. I think it's easiest to conceptualize it. All right, and then we also see that the pressure drops as you go further and further away from the aorta. Why would that be the case? Well, there's really two ways of thinking about this. One is mathematically, which I kind of prefer, but then two conceptually. So mathematically, remember that fluid flow is caused by change in pressure and limited by resistance. And so, right, if you were to manipulate this, you could say that therefore change in pressure is equal to fluid flow times change in resistance. And so as you get further and further away from the aorta, aren't you having more and more resistance, right? That's basically saying we're adding our vessels are being added in series, right? And then in individual sections, they might be in parallel, but the net result is you have one complete loop, and the sections of the loop are all in series. And so the further you get away from the aorta, the more resistance you have, and therefore, if resistance, excuse me, I misread this, yikes, I can't math. Delta P is Q delta R. If resistance is going to go up, then change in pressure is going to go up, okay? But what that means is that change in pressure is P final minus P initial, and so the delta, the difference between P final and P initial is going to be greater, which actually means that P final is going to be smaller, all right? So again, maybe the math way is like a little bit more confusing, but conceptually, right, if the resistance is increasing, the pressure drop over each section is going to increase. And so here's maybe another way of thinking about it, right? So this is our aorta, right? There's tons of high pressure blood because it's just come out of the heart, the pump, and then you have all of these resistors, the vessels giving us resistance, and then right in this next section you have less pressure, and we know that nature abhors a vacuum, right? And so you want to go from high pressure to low pressure, but it's going to be limited by this resistor. And then so then you think to yourself, well, wouldn't eventually this this chamber at P2 equalize with this chamber? Well, that would be true, but we have this pump. So these resistors are going to keep our pressures across different parts of the body from equalizing when in conjunction with the pump that is the heart. And so as you get further and further away from the heart with more and more resistance, you'll see that the pressure is going to drop overall. All right, so and that's what we see here. All right, that brings us to a couple of final things. Percentage blood volume. We have, let's see if we can get rid of some of these markings. All right, percentage blood volume in the different vessels. We can see that it is in fact greatest here in the veins, and that's because the veins have the highest compliance. So the total percentage of blood volume is the blood at each level dependent on the pressure and compliance of the veins. And so compliance is defined as the ratio in change in volume over change in pressure. And it's specifically change in volume produced by a change in pressure. And so it turns out that the veins can change in volume a lot for a relatively small change in pressure, so they have the greatest compliance, therefore they'll hold on to the greatest amount of blood. All right, so equation 5 is compliance. Compliance is equal to delta V over delta P, and then our final equation for this slide, which we get from our hose, is flow is equal to velocity times area. And so we saw that when we put our thumb over the edge of the hose, we decrease the area. Flow, right, you still have the same amount of fluid leaving the hose, so if Q is staying the same, then velocity must increase to compensate for a decrease in area. All right, so more about pressure. Specifically, one of the points that was brought up with is you have some high pressure in your the veins of your feet. And so the idea is that, imagine you're standing at the very bottom of a deep pool. You feel all of the weight of that water above you. And so the example that we typically give in physics is, say you have this nice water cooler, and, normally you go there to drink your water, but today somebody has poked a hole in it. And when the water container is still very full, the water shoots out really fast because the pressure at the hole is dependent on all of the water above it, right? And then, as it continues to leak, when the water level gets lower and lower, you see that the amount of water that's shooting out, it's not shooting out as far. Why? Because the pressure is less, because there's less water above it. So pressure of a fluid is also going to be dependent on the weight of the fluid above it and how much there is. And so in particular, you get this equation, which is pressure is equal to weight times height, and then you can take another version of that, which is how do you get mass? M times G is weight. You can calculate mass from the fact that density is mass over volume. So you can substitute M with density of the fluid times the volume of the fluid, and then this term times G, the acceleration to gravity. You get the weight of the fluid times the height overall of the column of fluid. All right, and so the purpose with this equation here, equation 7, is that the pressure, there's going to be a big pressure the further you get down in your body from all of the fluid being on it, right? The venous pressure is going to increase the further you go down towards your feet, right? And so that gives us a couple of definitions we need to know. Specifically, it's important to know this is why you have varicose veins often in the legs, because you have the weight of all that fluid pushing down. And so we said that veins have a high compliance. They're going to have a high change in volume for that greater pressure in your feet due to the weight of all the fluid above it. And so as a result, the volume of your vessels is going to increase, your veins are going to look extended, and they'll be varicose. All right, so then a couple of definitions that are worth knowing. Transmural pressure is the pressure across walls of vessels, and then perfusion pressure, which we'll look at, is the pressure gradient across tissue. So P perfusion is equal to the pressure in artery minus the pressure in veins. All right, so one quick thing about arterial pressure. The idea is that arteries are elastic, such that in between compressions during diastole phase, the overall pressure doesn't go to 0, right? When we measure people's heart rates, we're like 82, or blood pressure 80 to 120, but it's not 0 to 120, right? And so the idea is that in between compressions, some of that energy from the blood hitting the elastic part of the blood vessels is going to distend the blood vessels like a rubber band, and then during diastole, the rubber band, aka the artery, is going to snap back and return that pressure. And so you'll see that pressure will spike during contraction, but it never goes to zero due to the elastic property of the arteries overall. All right, but that gives us the idea that we have a mean and average arterial pressure, which is equal to the diastolic pressure plus 1/2 times the pulse pressure, and pulse pressure is going to be value given to us, but this should be the pulse pressure that you have at the peak of systole, and that's going to increase with the increasing stroke volume, and it's going to decrease with decreasing compliance, more elasticity, or less elasticity, more rigidity of vessels. All right, that brings us to laminar versus turbulent flow. Most blood is laminar flow, and turbulent flow is really this chaotic blood flow that we try to listen to if there's a it's what you would typically hear due to an obstruction or valve damage. We hear those as carrot cough sounds when oscillating the heart and mispronounced, I'm sure. But laminar flow is what blood flow normally is. It's smooth, predictable. Interesting point to know that at least was mentioned in lecture. The laminar flow velocity at the walls of the vessel is less than the velocity at the center. Why? Because you have friction with the walls of the vessel. In the center, you only have friction with adjacent fluid molecules. All right, and then turbulent flow is all chaotic. Mathematically, we can predict whether or not a flow is going to be laminar or turbulent based on properties of the fluid. In particular, we use Reynolds number. So Reynolds number is defined as density times velocity times diameter of the vessel divided by the viscosity. And then when you calculate it, if Reynolds number is greater than 2,200, we predict it would be turbulent flow for that fluid, and if it's not greater than 2,200, it would be laminar. All right, quick note about systematic versus pulmonary circulation. Pulmonary circulation, of course, is circulation to the heart. The idea is that higher arterial pressure is generally in systematic circulation generally is higher arterial pressure, and of course we know that flow is delta P over delta V. All right, so this is lecture 11, material blood flow regulation. The idea is that if you, so now we know some mathematical stuff about blood flow, we really have two main ways of regulating blood flow. One is locally with the endothelium, and then two is remotely using systematic, aka hormones, or neuronal control with the sympathetic or parasympathetic nervous system. All right, so let's start locally, talking about the endothelium. We're going to need to do some biochemistry a tiny bit, not really, but so our endothelium is going to be able to do local controls. We said it regulates smooth muscle constriction and dilation. This entire page is focused on dilation, so you're like, yikes, but it's not too bad. We're only concerned with nitric oxide. So do you remember nitric oxide, and you couple in your head nitric oxide vasodilation, nitric oxide, not nitric oxide vasodilation. All right, how does it do that? We'll go into, but nitric oxide vasodilation. All right, so here it is. All right, so endothelial cells produce nitric oxide as a gas, and it results in less calcium ions in the underlying smooth muscle. All right, so endothelial cells make the walls of our blood vessels. Deep to them is going to be our smooth muscle, at least in our vessels that can contract, right, which would be where we can control blood flow. And so you have nitric oxide coming from the endothelial cells going into our smooth muscle cells, and it will result in less calcium ions in the underlying smooth muscle. If there's less calcium in the underlying smooth muscle, that can mean two things. One, the smooth muscle isn't getting depolarized, therefore not allowing contraction. Or two, less calcium cells, and that's the primary thing. Or two, less calcium means less calcium for the sarcomeres, less calcium to bind to the troponin to move the tropomyosin out of the way to allow the actin to bind to the myosin to cause contraction, right? All right, so how do we actually produce nitric oxide? This is done by the enzyme nitric oxide synthase, right? And so there's really what's happening is we have L-arginine is being converted into L-citrulline plus nitric oxide by nitric oxide synthase, NOS. You have three different types, which we'll go over in a minute, but the main type is calcium dependent. Once the nitric oxide has been produced, it goes to our smooth muscle cell. The nitric oxide in the smooth muscle cell is going to stimulate guanylate cyclase and GTP to form cyclic GMP. Cyclic GMP is going to lower the calcium ion concentration in the cytosol, and then it's going to increase the potassium concentration in the cytosol. And remember that the net result of this is going to be a hyperpolarization of the cell. All right, if you're hyperpolarized, it's not going to contract as easily. You're going to, if you're not contracted, you're going to be dilated, right? Or if our vessel contracts, it's constricted. If it's not constricted, then it's dilated. So cyclic GMP is going to hyperpolarize the cell by lowering calcium ion concentration. All right, and then eventually, if we want to stop this process, cyclic GMP can be broken down by cGMP, cyclic GMP dependent phosphodiesterase, and that will break it down into GMP, and that's going to end effects of nitric oxide. All right, so nitric oxide in our head, vasodilation, produced by endothelium. Okay, so we can control the amount of nitric oxide produced in a couple of different ways. The first way is, and these are both stimulating, so increasing the amount of nitric oxide, flow-dependent vasodilation. And so the idea is that if you have say an increase in blood flow across a vessel, that's going to cause a lot of friction on the walls of the vessel, and the result is that more frictional force or shear force on the walls of the vessel due to the flowing fluid is going to release more calcium, and more calcium that the endothelial cells can take up, released by other endothelial cells, are going to stimulate calcium-dependent nitric oxide synthase, so CNOS. All right, CNOS being stimulated means more non-stimulated, which means more vasodilation. All right, the second way is receptor-mediated vasodilation, and so the idea is that there's a number of different endothelial cells or endothelial receptors will bind acetylcholine, bradykinin, substance P, I'm not sure exactly what that is, adenosine, and so if those can bind to the receptors in the endothelial cells, cause them to release more calcium into the vessel, into the blood vessel, this can be picked up by other epithelial cells, which will stimulate CNOS, which will produce more nitric oxide, which will do more vasodilation. Okay, so then what are the different types of nitric oxide synthase? We talked about CNOS, it's calcium dependent, it's going to do flow-mediated vasodilation, and receptor-mediated vasodilation, basically can stimulate by providing more calcium. And then there's iNOS, which is calcium independent, and this is stimulated by inflammation. And so the idea is that if you have inflammation, then ions will be stimulated, cause vasodilation, and then, like, what are the things that can help cause this inflammation? That would be bacterial endotoxins such as lipopolysaccharide, cytokines like tumor necrosis factor, aka TNF. All right, then our final isoform of nitric oxide synthase is N-NOS, neuronal nitric oxide synthase, and then basically the idea is that nitric oxide is actually used in the central nervous system as a neurotransmitter. All right, so we don't really have to be concerned with that one, but it's good to know. All right, and then we have different diseases that inhibit nitric oxide production, so they're going to inhibit vasodilation. That would be heart failure, hypertension, diabetes, elevated cholesterol, and elevated triglyceride levels. All right, and then different drugs, so C-dependent, cyclic GMP-dependent phosphodiesterase antagonists. So basically, if you attack the thing that's breaking down cGMP, then the net effect of nitric oxide is going to be maintained if you inhibit this guy that's going to break down cGMP. All right, and so this will prolong the effects of nitric oxide by preventing breakdown, and this will maintain vasodilation. I think Viagra was the example given to us in class. All right, so more local regulation, we have local vasoconstriction, and we just have, this is endothelin, which is ET1, and also produced by endothelial cells. It binds the receptors on smooth muscle. It causes calcium release from the sarcoplastic reticulum, and that's going to promote contraction. All right, and so this is produced by the endothelial cells from a molecule called big ET1. And so big ET1 is going to be catalyzed by ECE, which is this enzyme that's going to convert it into ET1. Then ET1 will bind to G-coupled receptors on the smooth muscle and then result in more calcium release from the sarcoplastic reticulum, and you get more contraction. But ET1 can be, or the formation of ET1 by the enzyme ECE can be stimulated by angiotensin II, antidiuretic hormone, thrombin, cytokines, reactive oxygen species, and then shear forces. All right, so then we have the concepts of what happens if you have say a blockage in the in the vessel, and then the vessel is suddenly not blocked for whatever reason. We have something called reactive hyperemia, and hyperemia means excess blood in the vessels. And so say you have a blockage and for a time you have no blood flow, then afterwards you're going to have an increase in blood flow due to an increase in vasodilators. All right, so reactive hyperemia is increase in blood flow after a period of no blood flow, and the thought is that the endothelial cells are continuously producing vasodilators, and these other vasodilators are H+, lactate, calcium, or potassium ion, carbon dioxide, adenosine, and then prostaglandins. And if there's a blockage, these continuously produced vasodilators are going to accumulate, and so they will produce their effect of causing dilation. And so after the blockage is passed, all of these accumulated vasodilators are going to have that vessel be dilated, and you have increased blood flow. All right. And so one of the interesting things here is that after you have increased blood flow, well, nitric oxide is going to be stimulated due to the shear forces on the due to increased blood flow on the vessel walls, and so more calcium will be released. Right. This is what we said happens in flow-dependent vasodilation. Friction along the walls of the vessel result in calcium being released, resulting in nitric oxide being released, which will result in vasodilation. And so nitric oxide will further cause more reactive hyperemia, excess blood in the vessels, by causing more vasodilation. So you can use nitric oxide blockers to counteract this. All right, then you have active hyperemia. Again, hyperemia, excess blood in the vessels. And active hyperemia is due to basically it's increased blood flow to an organ if the metabolic demands in the organ are increased. And so you can think of this as a pretty good thing, right? If you have more metabolic demands, say you're running a whole lot or something, your heart needs to pump faster, you need more blood to an area just because you're doing more, you're going to have an increase in production in our same vasodilators, hydrogen ion, lactate, potassium ion, carbon dioxide, adenosine, and the prostaglandins. Okay, and then this active hyperemia is the primary method of vasodilation in skeletal and cardiac tissue. All right, so you can think exercise for skeletal and then eating for intestines, and both will cause active hyperemia. All right, so increase in tissue metabolism is going to result in increased production in vasoactive metabolites, resulting in vasodilation. All right, and then our one example of vasoconstriction so far has been endothelin, ET1, which is going to be produced by endothelial cells and cause calcium release from the sarcoplastic reticulum, promoting contraction. All right, we're getting closer to done. Autoregulation is the ability to maintain a constant blood flow despite changes in perfusion pressure. What was perfusion pressure? That's the pressure gradient across a tissue. And so the idea is that, if you have an increase in pressure, then we know that fluid flow is caused by pressure limited by resistance. And so if pressure increases in the vessel, fluid flow is going to increase initially, but the body likes to maintain homeostasis, right? And so over time, the fluid flow will go back to the steady state values due to autoregulation. All right, so why would that be the case? And this is true within what we call regulatory range. So a certain blood pressure, so it's like, maybe 60 to 160, where, the body can get you back to regular blood flow as long as you're within the autoregulatory range of perfusion pressure. If you're beyond that, then there's not too much these mechanisms can do. But there's two different theories or hypotheses for how autoregulation works. The first is the metabolic idea, and so this is basically that increased pressure is going to cause that increase in fluid flow initially. That increase in flow initially is going to remove the concentration of the vasodilators that are constantly being produced, right? That's the H+, lactate, potassium, carbon dioxide, adenosine, prostaglandins. And as a result of that flow, that increase in flow, removing the concentration of vasodilators, you're going to have some vasoconstriction as a result, and this is going to decrease the flow and return your overall flow to the organ back to normal. All right, so metabolic hypothesis is increase flow, remove vasodilators, cause vasoconstriction, get flow back to normal despite the increase in pressure. All right, so then myogenic hypothesis is that the idea that when muscles constrict, it's based on the observation that when muscles stretch, it contracts. So if you have an increase in pressure, say in an arteriole, then the muscle around it's going to be stretched, right? There's high pressure in the vessel, and the muscle around it's going to be stretched. If the muscle's stretched, then it's going to mechanically contract, right? When muscle is stretched, it contracts. This is going to cause vasoconstriction, and it's going to return the blood flow back to normal, the net flow back to normal, right? Because flow is equal to area times velocity. If you constrict, this is going to go down, and you're going to have a net decrease in flow, or get it back to normal. assuming that velocity is staying roughly the same. All right, so basically that, you have this idea further that muscle can act as a pump in different parts of the body. So if you're running, then the contractions of your muscles are going to push on your veins, and by pushing on your veins, it's going to push the blood through the valves in your veins, and then when you relax, the valves are closing. And so the idea is that, if you're running, then the muscles will act as a pump and return the blood to your heart faster. Right? It's actually pretty cool because then you can exercise because of this mechanism that returns blood faster. If blood's returned faster, then it can be reoxygenated faster and then redelivered to the rest of your body, and you can continue running. All right, so mechanical effects of blood pressure, perfusion pressure, which we said again is pressure gradient across the tissue, can be calculated as mean arterial pressure minus the venous pressure. And then if the tissue pressure is greater than the venous pressure, say tissue pressure due to muscle contracting, as in here, then perfusion pressure is equal to the mean arterial pressure minus the tissue pressure. All right, then we have remote blood flow regulation. We have hormonal control, circulating factors, and we have neuronal control. Neuronal control, there's not too much to know here because we already pretty much know it, but for blood vessels, the parasympathetic is going to do blood vessels in the heart, genitals, and brain. We know that parasympathetic uses acetylcholine with the muscarinic receptors. It's going to act to increase nitric oxide. We have drilled into our brain. Nitric oxide means vasodilation. So parasympathetic nervous system will do vasodilation. Sympathetic nervous system is going to work on all the other blood vessels, including some in the ones that the parasympathetic nervous system works on, aka heart, genitals, and brain. And you can see that here, right? We have the blue is the sympathetic, and we can see that it's going to the blood vessels, it's going to the heart, and then we have the brain up here. All right, sympathetic postganglionic cells are going to have neuroepinephrine released onto alpha receptors, and alpha receptors on the vessels are going to cause vasoconstrictions. Alpha receptors, vasoconstriction. Alpha receptors, vasoconstriction. All right, it can also do vasodilation in skeletal muscle and skin using either acetylcholine or neuroepinephrine, but generally, if you were in that's in skeletal muscle or skin. Why would you want that in skeletal muscle? Well, sympathetic is, fight or flight. And so you would want to dilate the vessels in your skeletal muscle, right? So you can fight or flight. All right, circulating factors. So these are the, pump into your bloodstream to cause vasoconstriction or vasodilation. The main one is for vasoconstriction. This is the renin-angiotensin 2 mechanism. And so the idea is that there are 3 different causes that would result in this mechanism being triggered. The first is a decrease in pressure in the kidney, right? Why would, decrease in pressure? We'd be like, hey, that's not our homeostatic value. We want to increase the pressure. So trigger this pathway, right, to get back to the normal pressure. Then extra for whatever, an increase in sympathetic nerve activity in the kidneys, and then a decrease in sodium concentration will also cause this to occur. But basically, once this pathway has been triggered, the kidneys are going to release renin. Renin is going to convert the angiotensinogen released by the liver into angiotensin 1. Angiotensin 1 is going to be converted by the converting enzyme in the lungs to angiotensin 2. Angiotensin 2 is going to cause vasoconstriction. All right, so kidneys release renin. Renin is going to convert angiotensin to angiotensin 1, angiotensin 1 to angiotensin 2. Angiotensin 2 causes vasoconstriction. All right, and then we have another one which we love, which is vasopressin. Right, how can we remember this? Pressin, like pressin on vessels, vasos. And this is also called the antidiuretic hormone. So vasopressin, also called antidiuretic hormone, which we often write as ADH. This is released by the pituitary gland, and it's going to activate V1 receptors on smooth muscle and cause vasoconstriction. All right, so vasopressin, aka ADH, and then angiotensin 2 from the renin-angiotensin system are going to cause vasoconstriction. All right, then we have vasodilation from epinephrine. So neuroepinephrine releases circulating factors, right, not as neurotransmitters, by the adrenal medulla, right? Why would they release it? Because of nervous system stimulation, but still. So epinephrine released in the bloodstream will bind onto beta-2 receptors in vascular in different vessels and cause vasodilation. Remember, we said neuroepinephrine for alpha will cause vasodilation. And so epinephrine will bind to beta-2, causing vasodilation. However, if there's a lot of epinephrine, high concentration of epinephrine, that can bind to alpha receptors and cause vasoconstriction, which is what neuroepinephrine generally does, right? Neuroepinephrine through the bloodstream will bind to those alpha receptors, and they will cause vasoconstriction. All right, but beta-2 receptors are going to cause vasodilation with epinephrine. All right, so then one quick final thing, microcirculation in veins. We're familiar with the different types of capillaries: continuous venturated sinusoid, aka discontinuous, and are continuous as small pores only, no pores if you're in most of the central nervous system, right? You don't want pores in your, letting things in your brain. And then the fenestrated in the kidneys are areas with lots of blood flow, letting small molecules through. And then the discontinuous or sinusoids in the liver and allows larger materials to get through. All right, then we have our last 2 equations. Stirling forces, we're familiar with the concept of osmosis, right? If you have a high concentration of ions or materials in, say, like one fluid, and then you have a second fluid next to it, separated by a membrane, that doesn't have as high of a concentration, water is going to flow from into the compartment with higher concentration of material. And so the same thing is happening here. Stirling forces are describing filtration. So if you're in a capillary bed, going from capillary to, the venous end of the capillary bed, or venules, you have a couple things at play. You have the pressure from, the fluid flow overall, right? The pressure, fluid being pumped through the capillary, and that's going to decrease as you go along the capillary due to, more resistance, right? Resistance changes with length, drinking through a long straw versus a short straw. All right, so we're going to represent that as PC. Then we have the oncotic pressure. And so the pressure, right? If you have a lot of stuff, if you don't have very many things in the capillary side of things, there's going to be pressure drawing water in, right? Or if there's a lot of things in the capillary, there's going to be pressure drawing water in, so pi sub c. And then the same thing on the tissue side, pressure from the liquid on the tissue side, and then pressure due to concentration of material outside, the oncotic pressure of the tissue. All right, and so net filtration force is equal to the filtration forces pushing fluid out of the vessel and into the tissue, minus the absorption forces pushing fluid back into the capillary. Okay, and so you have P sub c, which is going to push fluid out, plus pi t, the oncotic pressure of t, depending on the concentration of material outside the vessel, minus P sub t plus pi of capillary. All right, and the net filtration is that filtration, it's a constant, filtration constant times the filtration force overall. Okay, we're just about done. One last thing, precapillary resistance. So if the resistance right here and the arteriole, which has got, the sphincters ahead of your capillaries, if you increase resistance by constricting, right, making a thinner pipe, thinner straw, if that resistance goes up, then the pressure in the capillary is going to go down. Why would that be the case, right? So here is precapillary and the arteriole, like right before the arteriole. If the arteriole gets smaller, right, then it's going to be harder for these guys to move through and increase the pressure over here, right? And so if R1 decreases, if the precapillary resistance right here, if our arteriole constricts, then the pressure over here is going to decrease because it's harder for the fluid to get into this area, into over here to pressurize it, right? And then in the reverse, if the arteriole resistance decreases, right, opens up, if this becomes really wide, well then it's really, easy to get all of the fluid over here, and this pressure is going to increase. Okay, all right, and I think that's just about it. Control of venous compliance. Basically, you can make veins. How can you change the compliance of a vein? We say compliance is the ratio of change in volume over change in pressure, and so a big change in pressure, veins have highest compliance because they have the biggest change in pressure, or biggest change in volume for the lowest change in pressure. Basically, vascular smooth muscle the activity of it can be changed by sympathetic stimulation or by circulating vasoconstrictors, and also by temperature. And then passive control is just change in body position will change the pressure in the vein and therefore change compliance in the long term. All right, so that's it. Good job, guys, and that was super long, but we're done.